we have a bunch of tools. Let me show you a simple example of what we can use these tools for. So we have this question. If we have a catalyst, an enzyme, that works really well, we ask a simple question, can we make it work better? So we took a very common catalyst, a very common enzyme. It's horseradish peroxidase, and this is used as a detector essentially a lot of times in biology because the substrate, if you put Amplex red, the enzyme acting on the substrate makes it start to it makes it into a dye molecule that fluoresces. It makes resolution. So this is commonly used as a marker in detection in biology. And in horseradish, it exists in nature. Nature has really perfected it over the years. And so we asked a sort of a silly question, could we possibly improve it? And the right way of doing that is to go in and understand the chemistry of this horseradish peroxidase and then understand really the details of the chemistry and do chemistry to improve it. We're not very good at that. That's not the way we did it. We did what nature did. We used what Darwin told us about. We used evolution. So remember, an enzyme always has a parent gene that codes for the enzyme. And so what we did, and Jeremy did that, was we took the parent gene and we reproduced the parent gene, but we made lots of mistakes in it. We made mutations in that gene. And then we did evolution. We just asked, okay, we have something that we've made a mutation. Is there some chance that one of these mutations works a little better? That's what evolution does. It finds mutations, random mutations, and if it improves, then it survives and it gets better. So we did exactly that. We used error-prone PCR. We used a bunch of other ways. Basically, we took the parent gene and make a library of mutated genes. In these days, this was done about 10, maybe 8 years ago. We weren't as good at manipulating things in drops as we are now. So we didn't know how to do the translation, how to express the gene from the how to express the gene into the enzyme. So we worked with Dane Ritropet at MIT, who could trick a yeast cell. He'd put this the parent or put the gene inside the yeast as a plasmid, and he tricked the yeast to express the gene on the surface. So it's surface display of that. And so now if you expose the yeast to the substrate, it'll turn the substrate and make it fluoresce. So here's a case where we took the parent gene, we put it on the yeast, surface display. All the drops that have yeast cells you see are bright because we're looking in fluorescence. So now we can do the experiment. It's a fairly simple experiment. We make a library of plasmids. Each of these have a mutated gene. We put one plasmid per yeast cell, one yeast cell per drop. We add the substrate, and we just look for fluoresces. If it fluoresces, we sort it out. But what's nice about this has a certain advantage. What's nice about this is that what's producing the enzyme now is a living object. So we can take these, and the ones that we sort, we can break the emulsion. It's just yeast. We can grow it up, and we can sort it again. And we can do this several times. So this is the experiment. We grew these yeast up, and if we used the wild type that always produced an operating enzyme, then this was the average level of fluorescence in the drops. When we first did this mutation, most of them don't work. Most of them, you're doing mutation and you're destroying the enzyme. You're just killing it. It doesn't work. And so they're dark, and only a few are fluorescent. So we collect them, we sort them, and then we grow them up, we do another sort, and ultimately we can get a bunch that really fluoresce well. Now we've really sorted out those that work well. And since they're yeast, we can sequence them, we can figure out what they are. And we did some estimates. We started out with a library of about 10 to the 7 mutants. And just by doing some sequencing and a little bioinformatic estimation, we figured we had a couple of hundred unique mutated enzymes that worked roughly as well. We set the bar really low. We didn't ask, oh, make it work 100 times better. We said, just, does it work? Can we see something? And we found a bunch of mutations. But we only found 100, roughly 110 to the 7. Not many, right? But then what we did was we took the best of those. We should have just taken all of them. We took the best 20. And we did a second round of mutation. And we did the same thing over again. And now, very rapidly, we found things that work way, way better. So what are we doing? Here's a simple way of thinking about it. Think of a fitness landscape, and the fitter the enzyme is, the higher it is. So nature has made this work really well. It's sitting on top of the peak, and we're saying now, please can we make this by making mutations. This is mutation space. Making mutations. Make it work better. Go to a higher peak. But the problem is most mutations take us into this valley of death. It takes us into this water. It just destroys the enzyme. What we did was we did one mutation, and we found those that climbed a little way up the valley. And then we did a second that climbed up to all the valley. So you can do this. You can do this normally. The problem is that before we had the microfluidics, the traditional way of doing it, the traditional way was you'd make the mutations and you grow up the yeast or the bacteria and you take a prick, you purify it, you look at it. So I estimated that in a quantum of time, what's a quantum of time? It's one graduate student lifetime. In that amount of time, somebody could do about 1,000 tests. I mean, it's a very lengthy procedure, 1,000 tests. But we had to look. We had to find not 1,000. We had to test 10 to the 7, and we only found 100. If you're testing 1,000 out of 10 to the 7, you have no hope of doing this. So this is called neutral selection, and this shows that this concept of neutral selection really works. In fact, you could do it with a robot. You really could. Here's why you wouldn't. If you did it with a robot, it would take about 5,000 liters of reagent, of medium, to do the sorting, just to do the sorting. For us, it took about 150 microliters. It would take you about 2 years to do the sorting. Just the sorting part would take about 2 years. For us, it took about 7 hours. Here's the killer. It would take about $16 million. And this is already a number of years ago, so with inflation, it's probably a little more. $16 million to the experiment. With the microfluidic, we tried to amortize things. We tried to be honest as we could. We figured it cost about $2.50. In fact, I showed this to my colleague, David Nelson, a theorist in my department. I showed it to him, and he said, you know, the most amazing thing about this is you don't pay your students very much. They work for 7 hours, you pay them $2.50. So I said, no, it doesn't include labor costs. So I do pay my students a little more. But they still work very hard, right? They work for 7 hours. No, I mean, this shows you why you don't want to do it. Okay, I think I could show you another example. Maybe I'll just show you one other example. This is a really new example of what we do. It just take a minute, then I'll shut up and I take questions. This is where we're really pushing. So if you do genomic analysis of cells, right now what you do is you collect all the information, and often you're doing a variety of different things, but often you're just trying to measure gene expression levels by sequencing the messenger RNA of cells. And the way you do that is you collect all the cells together, and you prepare your sample, and you use this wonderful instrumentation, the sequencing instrumentation. It gives you a huge amount of information. And in order to get that information, you collect a lot of cells, and then you sequence them. The problem is if you want to see very rare events, there's all kinds of noise introduced and all kinds of problems, but you're taking this huge number of cells and you're trying to see 1 or 2 rare cells. You can do it, but it's very difficult. And really rare things are basically impossible to identify. And particularly, you can't see cell-to-cell variations. So what you'd really like to be able to do is look at individual cells, but look at many, many cells at the same time. You can do that now. And I'll show you how we do it. I'll give you a very simple tour of how we can do that. Again, taking advantage of drops. The key is that we put cells in the drops. It's very easy. They're ideal sizes for the drops. How do you get a single cell in a drop? Well, you just dilute. You dilute down, so only 1 cell goes in maybe every 10 drops, 1 in 10. And then the number of cells, if you have 1 in 10, one drop has 1 in 10 drops has a cell. The number of cells where there's doublets, maybe a few % of the cells are 2. If it's a random process, it's Poisson statistics. So you just dilute, and you can do it very nicely. The problem is if you want to also add something else, and we want to add some particle that has some genomic material, the problem is how do you also do that? Well, we've developed a simple way where we can add a particle by using a soft particle. We can put 1 in every drop. And the nice thing now is that each particle, each drop has a particle and a cell. And on that particle, we put something that has a barcode, has genetic information that's unique to the drop. And so what we do is we encapsulate the cell, we lyse the cell, and we encapsulate the barcode, the particle, and that particle contains billions of copies of a piece of DNA that's exactly the same and is unique to that drop and that drop only. And so we take this, we capture the material, we create the library of, for example, the messenger RNA. We create the library, but it's now created where everything is labeled with an additional piece of DNA that's unique to the drop. Then we break the drop and we do our sequencing the way we normally do. I'm going to skip this as just showing all the details of the barcode. The important thing is that in the barcode, we have something that's labeled for the drop and therefore the cell. So not only do you read the sequence, but you read which cell it comes from. And there's a lot more details, but I won't go through the details of that. I don't have the time. It's not important. If you do this, you can collect a lot of information. Again, let me just skip over and show you the results. These are the kind of results that you can now collect. This is looking at the full expression level, the full transcript, the full level of messenger RNA for each and every cell for 920 cells measured simultaneously. So this is what you would normally measure, but you wouldn't know which cell it comes from. All we've done is we've added a label to tell you what cell it comes from. And this now becomes very powerful. If you want to look, for example, at stem cells and differentiation of stem cells, here's an example. This work, by the way, was done by Alon Klein and Mark Kirshner in a collaboration with us there at the medical school. Here they're looking at stem cell differentiation, and you can see just by grouping the transcripts, they can immediately see that after 4 days you see basically 3 different types of cells. Now you see multiple cells. They're starting to differentiate more. You can see different classes of cells just by looking at the gene expression level of them. So this is tremendously powerful now, and it's a very simple way of doing it. It's just taking advantage of these drops.